In Basics of Immune System Part 1, in the last video, we studied about the antigen presentation to CD4 positive T lymphocytes and antigen presentation to CD8 positive T lymphocytes. We discussed about the mechanisms of activation of CD4 positive T lymphocytes and CD8 positive T lymphocytes after antigen presentation. Today we are going to discuss about effector cells that are synthesized from CD4 positive T lymphocytes and CD8 positive T lymphocytes. So this video is about Basics of Immune System Part 2. Let's start Basics of Immune System Part 2. In this video, we will discuss about formation of effector cells from CD4 positive T lymphocytes and CD8 positive T lymphocytes. Dendritic cells, macrophages, and B lymphocytes are the main cells which present antigen to CD4 positive T lymphocytes and CD8 positive T lymphocytes. So that's why they are called professional antigen presenting cells. But infected parenchymal cells also can present antigen to CD4 positive T lymphocytes and CD8 positive T lymphocytes. For example, virus-infected hepatocytes can present antigen to CD8 positive T lymphocytes in association with MSC class 1 molecule, and these cells can present antigen to CD4 positive T lymphocytes in association with MSC2 molecules. Hepatocytes which are not infected, they do not express MSC2 molecules, but infected hepatocytes, they express MSC2 molecules and co-stimulatory molecules. So this is the reason why infected parenchymal cells can present antigen to T lymphocytes. What is MSC restriction? Antigen in association with MSC1 is always presented to CD8 positive T lymphocytes, and antigen in association with MSC2 molecule is always presented to CD4 positive T lymphocytes. So this is called MSC restriction. What are the effector cells of immune system? After activation of CD4 positive T lymphocytes, two effector cells are formed. These are CD4 positive type 1 helper T lymphocytes and CD4 positive type 2 helper T lymphocytes. In short, they are also known as Th1 cells and Th2 cells. CD4 positive Th1 cells help other effector immune cells. So what are the other immune cells which are effector immune cells? These cells are macrophages and plasma cells. Plasma cells are derived from activated B lymphocytes. Th1 cells, they help these cells, macrophages and plasma cells, to carry out their functions. So that's why they are called helper cells. After activation by antigen, CD8 positive T lymphocytes become effector cells, and the name of the effector cells is cytotoxic T lymphocytes. Now let's look at the names of effector cells. The effector cells which are related to CD4 positive T lymphocytes are CD4 positive type 1 helper T lymphocytes. In short, they are called Th1 cells. CD4 positive type 2 helper T lymphocytes. In short, they are known as Th2 cells, macrophages, and plasma cells. Cytotoxic T lymphocytes are the sole effector cells, which are related to CD8 positive T lymphocytes. Why Th1 and Th2 lymphocytes are called helper cells? These effector cells of CD4 positive T lymphocytes help other immune cells to carry out their functions. Th1 cells help CD8 positive T lymphocytes. They help macrophages in phagocytosis. Th1 cells help B lymphocytes to become plasma cells. B lymphocytes are also helped by type 2 CD4 positive T cells. So this is the reason why Th1 and Th2 effector cells are known as helper cells. What are the molecular details of effector functions of Th1 lymphocytes? Now let's study how Th1 cells activate other immune cells. We will study this topic in three headings. Number one, mechanism of activation of macrophages. Number two, mechanism of activation of cytotoxic T lymphocytes, and mechanism of activation of B lymphocytes. Now let's study how Th1 cells help macrophages in their function. Th1 effector cells use two signals to activate our macrophage. The first signal is Th1 lymphocytes secrete gamma interferon. Gamma interferon binds with its receptor present on the surface of macrophage. The second signal is Th1 lymphocytes induce macrophages to synthesize more CD40 co-stimulatory molecules. CD40 binds with CD40 ligand present on the surface of Th1 lymphocyte. In this way, with the help of these two signals, a macrophage is activated. Activated macrophages phagocytose target cells more efficiently. The target cells can be bacteria, cell debris, platelets, and red blood cells. Macrophages process antigen more efficiently and present the antigen to CD4 positive T cells more efficiently after activation. So in summary, Th1 cells help macrophages in their function in two ways. Number one, they help in more efficient phagocytosis by macrophages. The second, Th1 cells help in more efficient antigen presentation by macrophages by inducing processing of antigen and by inducing production of co-stimulatory molecule CD40. Activated macrophages mediate delayed type 4 hypersensitivity reaction. Macrophages change into epithelioid histiocytes, and these histiocytes form granuloma. Tuberculosis, tuberculoid leprosy, and sarcoidosis are some examples of granulomatous disease processes. Now let's study how Th1 cells, Th1 lymphocytes, help infected parenchymal cells like hepatocytes and CD8 positive T lymphocytes in their function. Th1 cells help CD8 positive T lymphocytes in peripheral lymphoid organs in the following ways. Th1 cells secrete gamma interferon, which binds to its receptor on the surface of antigen presenting cell and infected hepatocyte. Gamma interferon increases the efficiency of target cells like virus-infected hepatocytes and antigen-presenting cells to process viral antigens for presentation to CD8 positive T lymphocytes. So this is one antigen presenting cell. It can be dendritic cell or macrophage or B lymphocyte. Here you can see antigen is being displayed on the surface of the APC in association with major histocompatibility complex molecule, MHC molecule. So such antigen fragment is displayed on the surface after the antigen is processed intracellularly. Infected hepatocytes also can process the antigen intracellularly and display a fragment of the antigen on the surface in association with MHC molecule. In case of presentation to CD8 positive T cells, the MSC molecule is of type 2, MSC type 2. Gamma interferon also stimulates APC and infected hepatocytes to produce more co-stimulatory molecules on their surface. So this is gamma interferon. Th1 lymphocytes synthesize gamma interferon, and gamma interferon binds with gamma interferon receptor on the surface of APC. And Th1 cells also induce APC to synthesize more co-stimulated molecule, CD80. So during antigen presentation, co-stimulated molecule CD80 should bind to CD28, which is present on the surface of CD8 positive T lymphocytes. So in the absence of binding of these two molecules, CD80 and CD28, antigen presentation cannot occur. This illustration shows infected hepatocytes presenting antigen to CD8 positive T lymphocytes. Antigen that is displayed on the surface in association with MSC1 molecule binds with T cell receptor present on the surface of CD8 positive T lymphocyte. And co-stimulated molecule CD80 or 86 should bind to CD28 present on the surface of CD8 positive T lymphocyte. In this way, antigen is presented to CD8 positive T lymphocyte by antigen presenting cells and infected hepatocytes. And for this kind of antigen presentation, help from Th1 lymphocytes is needed. Without the help of Th1 lymphocytes, such antigen presentation to CD8 positive T lymphocytes cannot occur. Now let's study how B lymphocytes are activated or helped by Th1 cells. Activated B lymphocytes become plasma cells, and plasma cells synthesize different types of antibodies. This is the flow diagram which shows how B lymphocytes are activated. Antigen enters the body through the skin or mucosa. Lymphatic vessel takes the antigen to regional lymph nodes. In the follicle of lymph node, antigen binds to B cell receptor of naive B lymphocytes. Naive B lymphocyte means these lymphocytes have never been exposed to antigen before. Here on the right side, you can see the antigen is captured by antigen presenting cell, dendritic cell, and the antigen is taken to regional lymph nodes. In the paracortex of lymph node, dendritic cell presents the antigen to naive CD4 positive T lymphocytes. Naive CD4 positive T lymphocytes become Th1 cells, Th1 effector lymphocytes after antigen presentation. Now, two signals are required for the activation of B lymphocytes. Signal 1. Antigen is internalized by B lymphocyte and processed Antigen fragment with MSC2 molecule is displayed on the surface. Antigen plus MSC2 complex is presented to Th1 lymphocyte. Signal 2. Signal 2 of B lymphocyte activation. CD40 of B cell binds with CD40 ligand of Th1 lymphocyte. So in this way, with the help of these two signals, B lymphocytes are activated. Let's look at the illustration which shows the two signals which are required for the activation of B lymphocytes. The first signal is MSC2 plus antigen complex binds with the T cell receptor. The second signal is CD40 of B lymphocyte binds with CD40 ligand of Th1 lymphocyte. Th1 lymphocytes activate B lymphocytes. B lymphocytes become plasma cells, and plasma cells synthesize different types of antibodies. And these different types of antibodies have several roles to play. IgG is an opsonin. It binds with the target particle, which is to be phagocytosed. IgG and IgM can activate complement system. Complement system activation leads to inflammation and cellular cytotoxicity. IgG, IgM, and IgA, they play a role in sterilizing immunity and antibody-dependent cellular cytotoxicity. For details, you will have to read these individual topics in different chapters. Now let's study the functions of effector cells. Functions of effector cells of CD8 positive T lymphocytes. The effector cells are cytotoxic T lymphocytes, and these cells kill pathogen infected cells. For example, liver cells in viral hepatitis, malaria parasite infected hepatocytes. These cells also kill tumor cells, and this role is called tumor surveillance. In autoimmune disease processes, cytotoxic T lymphocytes are important in the etiopathogenesis of autoimmune diseases like type 1 diabetes mellitus. Cytotoxic T lymphocytes are important in the pathogenesis of type 4 hypersensitivity reaction, cytotoxic type. One example of this reaction is virus-infected hepatocytes are killed by cytotoxic T lymphocytes in viral hepatitis. These cells also play a role in the pathogenesis of graft versus host disease and transplant rejection. What are the functions of effector cells of CD4 positive T lymphocytes? The effector cells are type 1 CD4 positive T lymphocytes. In short, they are known as Th1 cells. These cells carry out their functions through other effector cells like B lymphocytes, plasma cells, macrophages, and cytotoxic T lymphocytes. Th1 cells activate B lymphocytes to become plasma cells, and plasma cells synthesize different types of antibodies. And in the earlier slide, we studied about what are the different functions of several antibodies. Viruses can be neutralized by antibodies, and this is known as sterilizing immunity. IgG antibody is involved in oxygenation of bacteria and phagocytosis. Th1 cells activate macrophages, and macrophages phagocytose bacteria. Th1 cells are involved in granulomatous inflammation against certain bacteria like Mycobacterium tuberculosis. Th1 cells are important in the etiopathogenesis of autoimmune diseases like type 1 diabetes. The other effector cells of CD4 positive T lymphocytes are type 2 CD4 positive T lymphocytes. In short, they are known as Th2 cells or Th2 lymphocytes. These cells can stimulate B lymphocytes, and plasma cells formed from such B lymphocytes synthesize IgE antibodies. And IgE antibodies help kill parasites by antibody-dependent cellular cytotoxicity, ADCC. One example is killing Ascaris larva in lungs. Th2 cells are involved in type 1 hypersensitivity reaction. It is also known as anaphylactic reaction or simply anaphylaxis. Type 1 hypersensitivity reaction can cause systemic or localized disease. Localized type 1 hypersensitivity reaction examples are bronchial asthma and hay fever. The examples of systemic type 1 hypersensitivity reaction is anaphylaxis after penicillin injection, anaphylactic shock because of different etiology. Cytotoxic T lymphocytes kill target cells by three mechanisms. The first mechanism is perforin granzyme pathway. Cytotoxic T lymphocytes make holes in the cell membrane with perforins and inject granzymes into the cell. Aspases are activated and the target cell dies by apoptosis. Death receptor pathway, the second mechanism by which cytotoxic T lymphocytes kill target cells. Apoptosis occurs by FAS ligand pathway. Cytotoxic T lymphocytes have FAS on cell membrane, which binds with FAS ligand present on cell membrane of target cells. Aspases are activated and target cell is killed by apoptosis. The third mechanism by which cytotoxic T lymphocytes kill their target cells, uh, TNF and gamma interferon, these are the cytokines which are secreted by cytotoxic T cells. These cytokines have anti-tumor and antiviral microbial effects. Okay, that was about functions of different types of effector cells and the mechanisms by which cytotoxic T lymphocytes carry out their functions. Now, memory T lymphocytes. After the inflammation phase is over, there is a phase of contraction. Approximately 70% of CD4 positive and CD8 positive effector T cells die due to apoptosis during this phase. The remainder of the cells enter the memory T cell pool. Memory T lymphocytes ensure a more rapid and effective response when the individual encounters a pathogen in the future. Thank you. That's all about the basics of immunity type 2. So you can watch my earlier video in which I have described basics of immune system. That was part 1, and this one is part 2. Thank you so much.